Assalamu alaikum. This is PharmaPhilia with a new topic of introduction of autonomic nervous system with the new course of ANS. So let's begin the video. Our body function is regulated and integrated by the two systems. And what are they? They are the endocrine and the nervous systems. What is the basic difference between these two systems? Endocrine system sends signals to the target tissues by varying the level of the blood-borne hormones, while in contrast, in the nervous system, more than 10 million neurons that constitute the human nervous system communicate with each other through a chemical mediator. So what is the basic difference? In the endocrine, the blood-borne hormones play the game, but in the case of the nervous system, human nervous system constitute from the millions of the neurons. So here the neurons play the game. They also exert their effect on the peripheral structure by releasing of neurotransmitter. So what is the basic difference between the neuropharmacology and the psychopharmacology? In the neuropharmacology, it deals with the drug that produce their primary therapeutic effect by mimicking or affecting the function of the ANS, or are called the autonomic drugs. These autonomic agents act either by stimulating the portion of the ANS or by blocking the action of the ANS. Here they are talking about the agonist and the antagonist of the ANS. While in the psychopharmacology deal with those drugs that affect the CNS. So here you can pick up the basic difference between the neuro and the psychopharmacology. In the neuro, it deals with the ANS, but in the psycho, it deals with the CNS. Neurotransmission process, drug affecting the CNS may act presynaptically by influencing the production, storage, release, and termination of the action of neurotransmitter. Other agents may activate or block the postsynaptic receptors. Deficiency or excess of the neurotransmitter may cause the multiple disease in human being. All these neurotransmitters combine with their receptors by producing their action, and regulate the physiological functions, but any form of deficiency or excess may cause the many diseases. For example, overactivity of DA in the mesolimbic pathway can cause the schizophrenia, while either cholinergic overactivity or dopaminergic deficiency may cause the Parkinson's. While in depression, there is a deficiency of the serotonin or non-adrenaline. In epilepsy, NMDA mediate overactivity or GABA underactivity seen. Patients with Alzheimer's disease have a significant loss of the cholinergic neurons. So by pathway you can link the disease. So you can easily link the effect of neurotransmitter to the particular organ. This is the anatomical layout of nervous system. Nervous system is further divided into the CNS and the PNS. PNS is further divided into the autonomic and the somatic, which is an autonomic is the topic of today's lecture. Okay, it is further divided on the three bases, that is anatomically, physiologically, and pharmacologically. So pharmacologically it is two types, cholinergic and adrenergic. Physiologically it have further two types, that is a sympathetic and parasympathetic, and anatomically craniosacral and thoracolumbar outflow. Why autonomic nervous system is named as autonomic? Because it is not under the influence of any other will. By contrast, the somatic fibers are often controlled by the will. Sympathetic and parasympathetic systems are the two main divisions of the ANS. So the point of difference between the sympathetic and parasympathetic is origin. First, origin, thoracolumbar, and the parasympathetic craniosacral. Distribution T1 to L3. Parasympathetic sacral 2 to 4, and cranial 3, 7, 9 and 10. Its distribution ganglia is limited to the head and neck, while in the sympathetic it is a wide, long and away from the organ. In the parasympathetic it is short or close to organs. Release neurotransmitter from the sympathetic system, majorly non-adrenaline and acetylcholine minor, and majorly acetylcholine in the parasympathetic system. Fiber ratio from the pre to post is 1:20 and 1:100. In the parasympathetic it is 1:1 to 1:2. Transmitter stability, diffuse for the y direction and is stable. In the acetylcholine, rapidly destroyed locally by the cholinesterase or metabolized by the cholinesterase. So the purpose of the sympathetic system is the stress condition, working in the stress condition and emergency condition, while as you know parasympathetic system is the rest and digest, so it will help in the assimilation of the food and conservation of energy. This is the picture of the sympathetic nervous system, distribution of the fibers to the effector organs. As you can see, the origin of the fiber and the distribution of the fiber to the effector organ. And here you can see the division of the parasympathetic fiber, its origin and its effector organs. So what is the basic difference between the synapses and the ganglia? Synapses is the junctional region between the two neurons where the one neuron will lay the impulse to the other so that the impulse is transmitted, while the ganglion is a site where the axon of the preganglionic fiber makes synapses with the neuron of the postganglionic. So what is the basic difference? In the synapses we are talking about the two neurons, while in the ganglion we are talking about the axon of the preganglionic fiber and the postganglionic fiber. So it is a basic difference between the synapses and the ganglion. Preganglionic and the postganglionic fiber in both the sympathetic and parasympathetic system, there are pre fibers, ganglia and post fiber. So the sympathetic and parasympathetic ganglia, most of the sympathetic ganglia are in the sympathetic chain. Some other ganglia are situated away from the sympathetic chain, while in the case of the parasympathetic, the preganglionic fiber make the synapse with the postganglionic fiber as the parasympathetic ganglia, because we are talking about the parasympathetic. The postganglionic fiber arise from the parasympathetic ganglia and terminate in the target cells. What are the cholinergic and the adrenergic fibers, and what is the basic difference between these two fibers? As the name indicates, cholinergic fibers are those which release the acetylcholine on the stimulation. And what are these fibers that release the acetylcholine on the stimulation are preganglionic fibers of the both sympathetic and parasympathetic, postganglionic parasympathetic fiber. Postganglionic sympathetic fiber that supply to the sweat glands and the piloerector muscles. Nerve supplying to adrenal medulla, skeletal, neuromuscular junction and some CNS neuron, while the adrenergic fibers are those that release the non-adrenaline on the stimulation. And what are these fibers? They are the postganglionic sympathetic fiber and the postganglionic sympathetic fiber that supplying to the sweat glands are releasing acetylcholine. So must pick up this point. Okay, next, sympathetic and parasympathetic target cells, sympathetic system, vascular, smooth muscle, visceral, smooth muscle, cardiac, and all these are the innervations that are innervated by adrenergic fibers and release the non-adenine. Why it is important to learn? Because by the innervation to the particular organ, you can link the effect of particular neurotransmitter to the effector organs and its effect. So it can link with the drugs also. Okay, this is the basic summarized picture of what I described previously. It is the somatic and the autonomic division. In the somatic, you can see its innervation to the skeletal muscles, where in the case of the autonomic have innervation in the heart, blood vessels, viscera, eye, and glands. Autonomic is automatic, somatic is under the control. So you can see its innervation in the skeletal muscles, and we move our skeletal muscles with our will, and it is in control. So what is a neurotransmission? Neurotransmission in cholinergic neuron involves 6 steps. The 4 is the synthesis, storage, release, and all these. Release and its attachment to the particular receptor, and then termination in its metabolism, uptake by the cell or the preganglionic fiber again. The recycling of the choline. Cholinergic transmission. Acetylcholine is the major neurohormonal transmitter at the cholinergic nerve. And in the adrenergic transmission, adrenergic neuron, the steps are the same that described for the cholinergic neuron, except that the non-epinephrine is the neurotransmitter instead of the acetylcholine. Neurotransmission takes place at neuron beat like enlargement that is caused as varicosities. All the 6 steps are the same, that is our synthesis, storage, release, and receptor binding of the non-epinephrine, followed by the removal of the neurotransmitter from the synaptic gap. And here you can pick up the point. The uptake 1 can be inhibited by cocaine and TCA, while in the uptake 2 it is inhibited by the reserpine, and the effector cells of the adrenergic neurotransmitter are alpha receptors and beta receptors. So that's all for this video. I will continue the next part in the next video. Till that, Allah Hafiz.